another protein here, and each can be activated by something that binds to it and sends a signal separately, but that the signal gets amplified. Basically, when something comes down certain signals and these proteins actually come together, they sort of bind or what we call dimerize, then the signal that they then send is greater than the sum of their individual wholes. The whole greater than the sum of the individual parts, which is what synergy means. And so I gave a one-word answer, and they thought about it, and they didn't want to like everyone kind of like it's like record scratch, like everybody's like, oh wait, they wanted to say no, that's wrong or whatever, but it actually summarized what the entire answer was reflecting. So either one kind of works, but you got to be able to back it up. Does that make sense? Okay. Any questions on the content that we've covered up to this point? We're going to launch from it, as I mentioned last class. So to launch forward on the congratulations that you finished the easiest part of class, and we will now start the next steps forward. We'll build cumulatively on what we know and what we talked about, and add a lot of new stuff. I wanted to finish off with that I never got to last time. What is
find some sources to send a Dr. Schultz so I actually have a presentation that works.
One presentation, one?
Yeah.
one thing I just wanted to show?
Do you want me to close the door?
Sure, if you don't mind. So this is just a little bit of follow-up neuroanatomy stuff, just in case it comes up in the rest of the class. I just want to show a couple pathways. So there's a handful of neurotransmitters, which are basically chemicals in the brain that are going to transmit signals from that presynaptic axon and send those signals to the postsynaptic axon. Those are chemicals, or not ions. We're going to talk about ions later today. Those chemicals basically are going to actually bind to some of those receptors and open up those postsynaptic cells for those ions to effect a change. Now, there's different types of chemicals that can be released. We call them neurotransmitters. Some of them might sound familiar to you, but I just wanted to just show you that they tend to have their cell bodies, or at least the big chunk of them, located in different parts of the brain anatomically, and send somewhat different pathways out. Okay, so the first one I'll mention is dopamine. Okay, it's a chemical that's highly involved in lots of things, from learning and memory to reward and pleasure-seeking activities. Okay, it's a neurotransmitter that's activated upon certain drug use, like cocaine, and then it's depleted thereafter that drug use, and leaves people in a big well without it. Okay, but the point of the figure is to show that a lot of those cell bodies are going to come from an area called the substantia nigra and the ventral tegmental area. You'll see that often abbreviated as the VTA, but it's in this midbrain area here, and you can see that the arrows are showing it projects to pretty much everywhere. Right? Okay. And so it'll come back down, it'll go out around, all sorts of areas. Okay? Now we can contrast that to a different type of chemical, norepinephrine, which we've already talked about a little bit from our rap videos. And the norepinephrine, on the other hand, is not going to come from the ventral tegmental area, but this time from an area called the locus ceruleus. Okay? So that's how you pronounce this word, is ceruleus. Okay? And that's going to be over here in this other area. Okay? Now notice that it's going to project to lots of other areas around the brain as well. But so if you look at these two figures, right, that we just talked about, the arrows are really quite the same, aren't they? Yeah?
Mm-hmm.
Okay, so that's not what the figures are showing. It's not showing where they project to. That's not the take-home point here. The take-home point is where does it start, not where does it go. Okay? So we're talking about different areas or cell bodies where they're highly concentrated. And then we can look at another one, which is serotonin. Now, serotonin, on the other hand, is often basically in what we call the rapid nuclei. The rapid nuclei. Nuclei is plural, because it's not one single nucleus of one cell, but the collection of many of them. And rapid, we pronounce that, the rapid nuclei. And it basically means red nucleus. So when you stain that in certain slides, you'll see that. Okay? So anyways, you're going to see that rapid nucleus back there, and it will also project to lots of areas. But again, the idea is that they affect lots of systems. A lot of these chemicals are used sometimes in the same cells all over the brain. Okay? So it kind of underscores this broader point about mucking with the system, whether it be by legal drugs or illegal drugs. All right? Lots of things mess with your system. This is going to change the chemical composition of your brain. So will that Mountain Dew? Lots of things do, right? There's this sort of straw man argument. You guys know what a straw man argument is?
Sounds familiar.
No.
So it doesn't have to do with Halloween or anything, but the idea is that, and you'll see this all the time if you guys are news junkies or like watching people debate things or whatever, they'll hold up what's called a straw man argument, which is basically they'll say something as if it's true, and then they'll go attack it and show that it's not true, and therefore their position is right. But you can't validate your position as right by virtue of tearing down a position that's not actually a real position. Alternatively, it's not really an opposing position. Does that make sense? So the reason I mention that is because you'll hear a lot of people say these sort of strawman arguments about something changes the brain, or this is exciting. Well, that's a strawman because the brain's always changing. Just about everything we do changes the brain. Actually changes the brain. Sometimes in a micro way, and most of the time not in a permanent way, but a change is a change, which means that argument needs to be reformulated to be a little bit more accurate. So when you might have something like caffeine, right, it's going to work on receptors, chemicals called adenosine, okay, which are going to have downstream effects on a lot of these different neurotransmitter systems. The reason is because of the broader idea to understand, big picture wise, how does the brain work, right? When we're using even so what is it, serotonin, so even when you use SSRIs for say depression, selective serotonin reuptake inhibitor, Prozac is one, there's lots of other ones, right? It's been selectively engineered and designed to inhibit just one type of reuptake inhibitor, okay? For reuptake pump, I should say. But even so, look at where the effects are going to be. Everywhere. So every time you're mucking with something, by intention or non-intention, even in the most precise bioengineering ways that we have available right now, you're still firing with a shotgun and not a rifle. So if you don't know what I mean by that analogy, when you have a rifle, you shoot one shot as a sharpshooter. It's like working with a scalpel in surgery. Very, very careful. One specific aim. A shotgun has a spray of multiple pellets because you use them to catch pheasants and stuff like that. And so the birds fly everywhere. You couldn't possibly hold it. Instead, you just get that once. You just hope that they spray and pray. So you spray a bunch of pellets, hope they hit some of those ducks. Okay, so that's kind of what you're doing when we're working with these chemicals, both medically or illicitly. All right, which presents a major challenge to us, right? Culturally, scientifically, medically, because there's some big challenges. How do we overcome these? You know, it's a deep question. That if any of you guys want to get into drug discovery or work on some of these elements, you guys would be the people pioneering the answers to those questions. But when we talk about these chemical signals, these electrical signals that are based upon the chemical signals, is what we're going to be talking a lot about this week. Keep that in mind. That as we're going to look at it at one cell connecting to a second cell for most of the week as a simple model, an example. But remember that these are models of big networks and systems. Okay? And we're not going to talk so much about the neurotransmitters, let's say, for the rest of the week. But I did want you to know, I meant to cover this during the anatomy section, where they're sort of located in, but also conceptually, it's an important principle of the nervous system is to understand that that's what we're doing. It's a shotgun or a grenade, just blowing everything up around it, rather than a pinpointed, targeted solution to, oh yeah, there's this one little thing we need to fix here, so we're just gonna, no. It just doesn't tend to work like that. It's gonna have widespread diffuse effects, which is why we have what you guys see and experience as side effects. All right? Get that big laundry list of side effects. You want to take this one drug that fixes a bellyache, and they're like, okay, now, just so you know, it might have side effects of.
Apple side effects are worse than that.
Yeah. So it's because of these kind of properties and principles. Okay. All right. Questions on any of that? All right. Now let me know if I'm going too fast at all today. Okay. So we're going to talk about neurophysiology today and action potential. So we finished off a lot of last class talking about electrical signals, and we talked about EEG in these electrical signals, and particularly, actually, do you want to grab one of those lights? Maybe it'll help people.
This one or the other one?
Your call. Okay. So we talked a lot about electrical systems for methods, EEG and things like that. And so these are examples of scout maps that you can get to plot representative differences in activity. Remember we talked about subtraction analysis? That's how you get these graphs.
Yeah.
So this color, right, which looks all nice and fancy and cool, it's merely a graphical representation of subtraction of this red line from this black line. Now, these are electrical traces that we talked about, which are ERPs, right? Okay, so remember we started off the first day of class talking about reductionism, talking about different levels of analysis. So we started the biggest, highest level of networks here, which are reflected by the same high-level network of these averaged hundreds of trials into electrical activity for single particular event types. But these, of course, are averaged from the EEG, the raw EEG during those trials, right? So those EEG traces represent the network of activity at one particular electrode site, right? Which means it represents the activity of millions of neurons in this particular area firing in synchrony, which means firing at the same time. So that tells us something new right off the bat that we haven't talked about a lot yet, which is that neurons fire. Okay, so we're going to spend a lot of time this week talking about. It's what I'm assuming that you guys have already spent a lot of time this weekend studying about, and hopefully you'll be spending a lot more time after this week studying about it too. Okay, so these millions of neurons are firing in synchrony at this area right underneath the scalp by this electrode site. And we're going to start talking about reducing that more and more to away from millions and millions of neurons, but how does one neuron fire and why does it fire? Okay? So we're going to start from these high levels. We're going to dispense with this and we're going to start looking at some very low level activities. Okay? So when we talk about neurophysiology, we were talking about essentially the study of this electrical, or the activity or the functional elements of neurons. And so we're going to start with this very basic model. So this is a petri dish, and this is our model, sort of pyramidal neuron. Okay? We have this nucleus, the dendrites, the axon, and the axon collaterals. Okay? Now, what we can do is we can stick some electrodes right around it. Okay? Now, see how this electrode is connected to a computer. Okay, the computer is a fancy little abacus. It can count stuff. In this case, it's counting your charge. Okay, so charge is basically measured in units of voltage, which represents actually the separation of charge between two areas that you measure. Yes?
How small are those little electrodes?
Incredibly tiny. Incredibly tiny. Yeah. And so we actually have two electrodes in this setup. We have a reference electrode, which is in this, we're going to call an extracellular bath, okay? Essentially a fluid that the neuron is resting in, okay? And we have this electrode, which is also not yet in our neuron. See how there's that little space in between them? So if we just measure this, what do we get?
It should be neutral.
Hmm?
Well, if it's far enough away from the neuron, it should be neutral. It would just be in the extracellular, right?
Yeah, so don't tell me what it should be. Tell me what it is.
Where is it?
You just follow the line. This is
I
measuring
can't
here, it's connecting here. What does it show?
Oh, I couldn't see the thing behind it. The one that says outside axon. So it's right outside the axon?
Well, yeah, remember we said it's not yet in. It's still,
Okay.
It hasn't.
There's no charge for it, right?
Yeah, it's zero. So we have a zero measurement. So our y-axis is volts, but since they're tiny, they're millivolts. Okay? So it's just hanging out, chillaxing by the pool, actually in the pool.
So it is
And-
neutral then, right? Is it?
Yes, of course.
Okay.
Yes, of course. So we have zero. Zero is a good measurement of neutrality. And we can actually see it. So it's showing us this in the picture. So we're walking through the actual figure and breaking down what it means, what it's telling us. Okay? And when we zoom in on that, this is our axon. Okay? Now this deep orange is the inside of the axon. Okay? This light orange is a layer of essentially the membrane of that axon. And the outside is, of course, our extracellular bath. We call it the extracellular, the outside of the axon. Okay? Now what we'll find out is that there is actually a separation of charge. And we'll understand why as we move into more detail. But we start off by introducing this idea that the inside of the cell is negative and the outside of the cell is positive. Now it's relative, but here, this little tip, okay, that represents this electrode needle. And as you wonderfully just said, it has not yet permeated the axon, has not yet permeated the cell, right? So if we ask what it's measuring, it should be neutral, because this is in the extracellular bath, and our reference that it's compared to is also in the extracellular bath. So if you subtract two things with essentially in the same solution, what would you get?
Zero.
Yeah, of course. That's why we have that. Now we're going to take this next step and we're going to understand what happens. Now you take that electrical needle, okay, you inject it into the cell. So now you're actually measuring. Remember, voltage, the units of voltage represents a measurement of the separation of charge between two locations, two sites that you're measuring. Okay? So we're measuring one site here and one site here. Now that site is inside the cell. Okay? Now remember, this stays here that we've already identified as being positive. But now we've already identified the inside of the cell as negative. So you subtract the positives and the negatives, and what happens is that when you stick that measurement device into the inside of the cell, okay, look at, we started off resting at 0 here, right? It enters the cell and it goes whoop, our measurement goes all the way down to -60. Okay, now we can actually detect a separation of charge, or a difference in charge. That's separated by virtue of our membrane, right? Isn't the membrane the thing that's separating the pluses and the minuses? Yeah. Okay. So that's a really basic, fundamental idea that we're going to build from. You guys feel with that? Questions? All right.
Question.
Yes.
So is it always resting at 60, -60, or is it like?
Good question. It is absolutely not always resting at 60. In fact, every neuron is like you guys, and that is a special snowflake, and unique, and special in its own right. And, you know, they're different, right? We're all different creatures of nature. We have different biology, different chemistry, right? We're unique little things. And so we have variance, right? Do all of you guys like the same flavor ice cream? Probably not. Do all of you guys rest at the same attitude level and mood? Probably not. except when class is over. But I don't know. Maybe one or two of you actually like class, and maybe you might be sad when class is over. I don't know. I don't know if I'm that lucky. But you know, but you get my point. So the neurons are going to have that individual variability too, which means that we call this the resting membrane potential. Okay. Yes.
And then it's usually always negative because you're subtracting it 0 from that.
The outside measurement, yeah. Because what else would you measure it to?
That's true.
You know. So that's kind of your reference, your control measurement. You know. So if you want to know, if I asked you, is it hot in here? Is this room hot or cold? How can you answer it?
Yeah.
Yeah, you tell me it's a stupid question. Hot compared to what? Cold compared to what? It's both right now, right? It's hot compared to Antarctica.
Yeah.
You know, it's cold compared to Death Valley. Or just outside.
Just outside, yeah.
Which might not be very different. Right? But you see what I'm saying? So every type of measurement, to make a real scientific measurement like that, is actually requiring a relative judgment, a relative decision to be made. By relative, we mean it has to be compared relative to something else. And so, totally. So this is our resting membrane potential. Every cell is going to be a little bit different.
So it could be like a little bit higher and a little bit lower.
It could be big time lower or big time higher. It could be lots of different things. Yes.
So it can be positive as well when we're doing this, or not so much. Just
if it's
between 0 and.
If it's positive, it's probably a heart cell that is in a prisoner in the lethal injection chamber of prison.
Yeah.
Seriously, because that's how they stop the heart in lethal injection when they kill people with potassium or something. Potassium chloride.
Yeah.
Is you hold, and we're going to find out about this today in terms of why. You hold this you give them a chemical that puts that resting membrane potential here. That means that those cells can no longer fire. But what happens if your heart cells can no longer fire?
Stop speaking.
Yeah,
So you die.
it's a morbid situation. It's very dangerous. It's why it's a poison. So they're generally not going to be positive, but at a technical level to your answer, it's possible that they could be that way, but it's just
Not
very
really
long.
going to happen naturally for what we're going to do.
Yeah, for the most part. So they tend to be negative, and they ought to be negative. And but that being said, you'll find that they generally are around here. So most textbooks you'll see will give a representative figure, a representative value. Sometimes it's negative 60, other times it's negative 65. I've seen textbooks show negative 75. Corey, your question? So yeah, good question. But there's gonna be some variance.
I think I saw in a video it said negative 55 or something.
Yeah, it could be, and measure any of us, we're all gonna be a little different on something.
So what number do you want us to remember for you? Negative 60, negative 65?
I mean, I would say the same kind of thing that I think I've been alluding to for most of the class discussion of some of these questions about the questions and answers, which is that I don't want to do the high school thing of telling you to just remember a number, a fact, a date, or a name of this. You know what I mean? Just
yeah. Okay.
I want to be. I would hope it could be understood, and whatever number you want to pick, be able to explain what we just talked about as to why you picked that and why it might not matter. You know what I'm saying? So, negative 60, negative 65, you'll probably see me use that a lot.
Okay.
It's usually pretty normal, that's what people use, but if you want to choose negative 64.7827, or negative 55, that's fine, but, justify it. Why? Be able to communicate with people. You guys might be teaching this later, or tutoring people with this later. I don't want this, rote regurgitation of, here's a magic number, and why? I don't know, because it's magic. This is, no. Yes, you see what I'm saying? Okay. So we've got the separation charge, and we're going to talk about some terms we'll introduce. We will rely upon them as we move forward. Basically, anions and cations. Okay, so these positive and negative little signs, the pluses and minuses. Okay, the pluses basically refer to cations. Okay, and anions are these negative ones. Okay, so anions just mean negative, cations mean positive. Okay, I don't remember however you want to remember it. A little plus sign has a T in cations, so plus sign is kind of like a T, you know, or maybe you're like a crazy cat lady and you really love cats and they're really positive for you.
Happy and onions make you cry.
Yeah, yeah.
Onions make you cry.
Onions make you cry. Cations make you happy.
But they both have ions.
So, whatever little ways you want to remember that. Know that, and these ions, okay, they're just little elements, right? Things like sodium and calcium and potassium. They're basically hanging out, and they're dissolved both in the extracellular fluid, but also inside the cell as well. Now, they're hanging out and dissolved in the cell and outside the cell in different concentrations. Okay, so there's different relative proportions of them. Make sense? Okay. And we will get into this a lot, but remember we talked about this membrane here? Okay? Okay, that's going to separate the outside from the inside, and we're going to call it a lipid bilayer. Okay? So lipid means fat, because it's made up of some fatty substance, and bilayer means two layers. Right? Bicycle. Two cycles, two wheels on your bicycle, as opposed to a unicycle. Biplane, which means two wings instead of one wing. Of course, they're stacked on each other, by the way. Biplanes have two wings, right? One on top, one on the bottom. You guys remember those old planes? Okay. What else? So yeah, two layers, and we're going to zoom in on this as we go along and take a look at those two layers. So you're going to see those words used a lot. Okay? Okay, so we just talked about a resting membrane potential being somewhere between -50, -80. And remember that it's negative because it's reflecting that the cell is more negative than the outside bath, which is more positive. Okay? Okay, so we're talking more about these ions, and they are going to travel across our lipid bilayer, this fatty membrane, and they're going to go inside and outside of our cells by virtue of channels. Okay? Think about it as bridges across rivers or tunnels between. I used to teach in New Jersey, so I could talk about the Lincoln Tunnel between Jersey and New York. That doesn't mean a lot for you guys out here, but the idea is that we can have ion channels, and they're going to be proteins that span across the membrane, just like we started off class today talking about, right? This is our membrane. We have a protein that goes across it from the outside to the inside, okay? And those proteins are going to form channels, little tunnels, that allow the ions to travel, okay? They're going to pass through. Now, some of these ion channels are just open all the time. It's just open and free, and people can migrate however they want, or ions can migrate however they want. But other, and that helps to sort of set a little bit of the resting membrane potential, because those ions do travel. But most of the ions, channels they're going to be gated, they're closed, because there's a gate that keeps them closed, right? We need boundaries. Boundaries are very good. Okay, so we have these barriers, we have these boundaries. Okay, and they can't allow the ions to pass from the outside to the inside or the inside to the outside. Okay, and that might sound mean and callous to our friendly little ions who just want to be free and roam across the range, but we'll find out that without that basic discipline in the cell's membrane, we would not be able to live. We would not be able to sustain life because cells would not be able to fire. So that simple separation of that charge, that simple barrier of these gates on the ion channels, okay, that allows every function of life to continue to occur. Okay, and just like any gate, think of like a castle gate, Game of Thrones or something, right? The gates can open and they can close. All right. So you have a drawbridge that opens or closes. You have a gate that opens or closes. You can have a channel that opens or closes and lets things come in and out. Okay. And as I mentioned, some of those channels don't have a gate; they're open all the time. We call them passive channels. Okay. And let's see here. And but the passive channels can be selective to only certain types of ions. Okay, so different ion channels will only allow certain ions to pass through, because certain ions have bigger and smaller diameters. Okay. Okay, so that's kind of what we refer to as selective permeability. Okay, and that some ion channels allow some ions, such as potassium. So if any of you guys don't know, K is the elemental signal for potassium. Na is the elemental signal for sodium. Okay, it's called selective permeability. Any questions? Okay. Here's a picture. Kind of says the same thing. Okay, so we have different channels here. Okay, different colors. Okay, so red here looks like it's for potassium. So here we have potassium coming out. Okay, and we have another potassium here. It looks like it's coming in. This is an open channel. It's passive. Okay, this looks like a sodium channel. It is closed. Okay, so what we call gating. Something needs to open it. You need a key to unlock the gate. Okay, those keys tend to be those neurotransmitters. Okay. Now we'll come back to that a little bit later. Here we have a sodium ion. See how it's a bit wider of a radius than our potassium? Remember I said they have different sort of widths and diameters? So some can pass through those tunnels and others can't. There's selective permeability. So you have sodium trying to get in. The bouncer's saying nope. You can't come in. You don't have ID. It's true. You need ID to come in. Paid for college, but being a bouncer. Jersey Shore, so do a lot of IDs. And I actually checked Bon Jovi's ID. Hard him. Didn't think it was him. Are you Bon Jovi? Like, yeah. So, let me see something. Seriously? Yeah. I was expecting like a stage name and then like his real like name like John Q. Public or something like that. But it really said John Bon Jovi on his ID. It was really cool. He got some big beard and that's fun. But he kind of tried to keep a low profile in the bar, but the word got around, and about 3 minutes later, the girl started screaming, and he had to leave. But anyways, the bouncer's rejecting that, sending him back out, right? You need ID, you need a key to unlock that door. You need something special, some pass to allow that to open, okay? Again, we have a passive, open potassium channel, and here we have a closed sodium one, okay? Now, we also see that this is a very illustrative graph because it shows us several other things. One big thing it shows us is the difference in concentration. By concentration, I don't mean that they're very focused. Okay, they're not trying to study for the test. They're concentrated in terms of a chemical phrase of being closely together in a certain area versus another certain area. So in this case, we separate that by inside versus outside. And we can see that sodium, okay, there's a ton of sodium on the outside of the cell. Lots of blue bubbles here of sodium ions. There's not a whole lot of them, relatively speaking, on the inside, right? Now we see that that's also the opposite for our potassium, right? So potassium is the opposite way. There's a lot of potassium inside. There's not a lot of potassium outside. Look, there's only 1, 2, 3, which is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Big difference, right? Okay, so they're automatically an opposite, what we're going to call a concentration gradient. There's a gradient from inside to outside. It's a really important term. Know what a concentration gradient is. because it's going to be a driving force. Okay. Now there's also chloride. Now chloride's an anion. It's a negatively charged ion. So sodium and potassium we've just been talking about, they're positive. Chloride, on the other hand, is negative. That's the Cl and the minus sign. Okay? Now there's a lot of chloride outside, not a lot of chloride inside. Okay? Alright, now what else do we have? We have a little bit of calcium. Okay? But there, yes sir.
Are there generally all the ions in the extracellular fluid of every neuron, or like certain neurons more specific to having particular ions on the extracellular fluid?
Good question. So the neurons can't necessarily control what's in their extracellular. So the extracellular fluid is pretty much what you might consider otherwise generically just brain. Like the fluid in the extracellular matrix, that generic word brain. Whatever else is out there. And that's going to be relatively constant throughout the brain. Now, you're going to have local changes, which I think is what you might be asking about, right? And some of those local changes are going to be local to certain areas that are firing, which all areas are firing at different times. And so I guess now I'm starting to say the opposite, in that since they're firing, all the areas are being used, you're going to have these exchanges happening of the ions, which mean locally around different neurons, you might have different extracellular concentrations of things. But it's a pretty microscopic. Oh, okay. I thought I was saying something profound or something. Wow, I thought that only happened in the movies. I didn't know the music really came. It's pretty cool. No, I think that for the most part, we model it as being generally the same.
Okay.
Wherever. At the local level, it's pretty microscopic. Yeah, they're going to be a little bit different here and there based on what's being used and replenished and all that kind of stuff. It would be a strongman argument for me to say that it'd be the same fundamentally across the rest of the brain, because of course we know that there's going to be changes happening. I don't know if that was too confusing of an answer to your question.
No.
For what were our purposes, always, particularly for this class, this is just the outside of the cell, and that's going to be the same wherever. For the inside, it's typically modeled as the same, most neurons anyways. Again, they're going to vary slightly, but for the most part, this is a pretty good representation. And a lot of this was discovered by two guys working at a place called Woods Hole Marine Biological Laboratories, which is in Cape Cod, in Massachusetts, south of Boston. And they got the Nobel Prize for discovering these elements of how the neuron's functioning, the relative concentrations, and how these different concentration gradients, together with what we'll look at next, which is the electrical gradient, how they work together to allow cells to actually fire, which allow us to live and be alive. So this stuff might sound simple or rudimentary or obscure, I don't know, but it formed the basis for the Nobel Prize work and everything about our nervous system. So we're moving into that. So we also have calcium, very little bit of calcium outside, but nearly no calcium inside. So that's a big thing to remember too, because we're going to come back to that later. And then what's keeping the cell negative, really, right? If there's not a lot of chloride in here, if there's more chloride, which is negative on the outside than there is on the inside, then why is the cell negative? Yeah, so a lot of the proteins that are inside the cell, the nucleus and all that stuff that we looked at in that first class we did on sort of basic neurobiology, all those proteins actually have negative charges on them. That's helping to keep that cell resting at a negative level. Okay? All right. So we talked about these different forces. Hopefully you guys know what the force of diffusion is, right? You pour a bunch of chemicals in